Hello student, this is Dr. Faizan Mirza. I'm discussing some questions related to genetic technology and selection and evolution with you in this video. So this question I've taken from October-November 2019, variant 42, and this question basically deals about a stickleback fish. They have just named this. They are saying it's having two distinct forms. Two distinct forms mean that this question refers to polymorphism. Polymorphism is basically occurrence of more than one phenotypes of an organism in a particular environment. So the saltwater form and freshwater form. So there are two type of forms here. The larger freshwater form is thought to have evolved from a smaller saltwater form. So this information is given to you. Always keep in mind that the information given in the first part of the question and the initial part of the question, it's always very relevant to what you need to address later. The questions deal with how you actually handle the comprehension skills. So your comprehension skills must be very relevant and you should be able to recall what you already know, and you should be able to apply to what's basically shown to you in the case study in this question or in whichever question they basically put forward to you. Now, the question continues. Both forms of these fish, they have armor plating on each side of the body. The plates are made up of bone and contain high proportion of calcium. Now, this already tells you that there's a high proportion of calcium. So you can just try to think what will be the sources of calcium and how can the fish be getting a lot of calcium when it's living, for example, in freshwater or in salt water. So obviously it will be relying on the calcium availability in the environment, whichever prey that it will be preying upon. Now they are saying that there is a gene EDA which codes for a protein which is involved in the formation of armor plates. This gene is having two alleles, low armor and high armor. They have mentioned this to us. Now they are saying three main morphs of armor plating have been described. So this is a single gene with two alleles, high armor, low armor. Complete morph armor plating are those which are found mainly in saltwater. So you should just link this initially that this is in saltwater. It is having many plates from head to tail covering most of the body. That obviously means that it will need a lot of calcium to make sure that entire body is covered with this armor. It provides defense against large and predatory fish, limits the growth of fish. So yes, the growth, the size stays limited, and this provides a barrier, you can say, against predatory fish, so it doesn't get preyed upon very easily. Now there are partial morphs as well, which are partial morph armor plating, and these are found mainly in freshwater. You can just say that okay, these are the ones in the freshwater. They have their number reduced of the they have reduced number of plates to cover only part of the body. So obviously they will need lesser calcium as compared to the ones which we have discussed earlier, which need more calcium. The low morph armor plating is found mainly in freshwater, has very few undeveloped plates, no body cover. So this no body cover one probably will be needing no calcium because there's hardly covering no calcium, at least not for the armor plating, because it's not having it. But yes, it will be susceptible to getting preyed upon because the body will not be covered, and it can grow to a larger size as well. Why? Because the body will not be covered in this armor plating. Explained by the variation in armor plating in stickleback fish can be described as discontinuous. Discontinuous basically means that there are discrete values. Discrete values, these are groups, discrete categories. There is one category, there's a second category, there's a third category, and each category is having a distinct phenotype, and there are no intermediates in between. So for example, high armor, or you can say partial morph, or you can say low morph, they all are in discrete categories. They are three categories. So the variation of this fish is an example of discontinuous variation because only one gene is controlling armor plating, because one gene is a condition for discontinuous, because in continuous variation we have polygenes. So you can recall the concept of polygenes for continuous variation, whereas for discontinuous variation there is one gene controlling one trait. So there are three distinct categories. There is no range, there are no intermediate phenotypes. You have to have two marking points for that and to explain the command what is explained. In 1982, they are saying that this lake the entire freshwater fish population was accidentally destroyed by humans. The freshwater that was destroyed by humans, which one would this be? So freshwater ones are these. These were the ones that they got destroyed because they are the ones that are found in freshwater. Okay, so they were accidentally destroyed. In 1990, a new population of fish was found in the lake. Most of these fish had armor plates from head to tail on each side. So in each side means they are fully covered. So that's why these new fish have armor plate from head to tail on each side despite in fresh water. So we already know that fresh water is supposed to have partial and low morph. This information is given to us. and saltwater ones are the ones which are complete morph. But this is kind of an evolution, which they are saying that since this was destroyed by humans, now the new population is the one which is supposed to be in saltwater is actually coming in the freshwater. So why is that so? So the freshwater habitat actually got colonized by complete morphs. So it got colonized by randomly. Since they have armor plates, they survive the predation from the predatory fish of freshwater. So this predation was averted. As a result, probably a mutation might have occurred in these fish, which allowed them to have the armor grown from head to tail. So you can state either reason. You need to state just one reason because this asks you for 1 mark answer. Further, they say from 1990, annual sampling took place in the lake. Each year showed a reduction in the number of individuals with complete morph plating from head to tail each side. They took place in relatively short period of time. 1990, 1983, 96% fish had complete morph. 1993, 13% had complete morph. So you can see the complete morph actually decreased. So why it decreased? Because we know that complete morphs need a lot of calcium. This information was already given to us. So we need to relate this to what is happening here. So we can go back and we can just annotate this here. This is what I do. I prefer annotating the question when I'm answering it. So it becomes easier for me to just keep on referring back to what I need to answer. You can even underline these parts and these statements so that you can keep coming back to this information when you are answering the question, because this initial information is already always very relevant for answering any question. So coming back, what our answer will be, how explain how natural selection occurred in this new stickleback fish population. Complete morphs have armor plates of calcium. This limits the size of this fish. From 1919 to 1993, the fish might be decreasing. They might be decreasing availability of calcium in the lake, or the number of predatory fish would have decreased. Now, if the number of predatory fish would have decreased, this stickleback fish would not need a lot of armor because the predatory fish would decrease. This armor synthesis would cause loss of energy and in the complete morphs, while other two phenotypes would have a selective advantage in both cases because they will be having more ATP available for growth and for all these processes because they are not wasting energy in synthesis of the armors. Further, you can say that these two phenotypes, so the other two phenotypes could grow larger, they could grow in size, they could lay more eggs, they increase their proportion in population as compared to complete morphs. This change in allele frequency is an outcome of directional selection. Yes, this is directional selection occurring. What is directional selection? Directional selection is that in which the population shifts to a new mean. So this dashed line, this dashed here, dotted curve actually is a directional selection. You can see that the directional selection occurred here. So if I say if I take this is you can say complete morph. This is complete morph. You can say this is partial, and this is incomplete. So if in this case, what's happening? So in the initial population, if complete morph was more, so now this population is moving to a direction selection, and you are having more of partial morphs and more of incomplete morphs. The next question, again from the same paper, this question, October, November 19, variant 42, this takes into consideration therapeutic proteins are used to treat diseases. The first purified therapeutic protein used was insulin in 1922. This extracted from animal pancreas since 1882. Most insulin has been made recombinant DNA technology. Explain the advantage of producing human therapeutic proteins such as insulin by recombinant DNA technology. So you just need to describe the process in which the recombinant DNA technology is used. So human proteins produced by recombinant DNA technology would have been identical in shape to the naturally occurring protein. This allows the functioning protein to be produced. In this way, the function stays optimum. There is no allergic reaction of such proteins because the immune response does not recognize them as a foreign antigen. The cost of production is lower as compared to protein isolated from other organisms. There are no ethical or religious objections on these as compared to taken from other organisms. Calcitonin is a small protein hormone consisting of 32 amino acids. One of its functions is to inhibit the activity of cells called osteoclasts that break the brown tissue. So we know that they are saying that there are cells called osteoclasts that break the bone tissue. Calcitonin has been used as a therapeutic protein to treat osteoporosis. Osteoporosis is a disease in which too much breakdown of bone tissue occur in old people, leads to reduced bone density and a risk of broken bones. Now part B, figure 3.1 shows to you the position of these amino acids. 32 amino acids are shown to you for human and for salmon. You can see that all these amino acids are listed to you. The positions are mentioned. So on first position, human and salmon, they both have the same amino acids, same. And on the third position, they both have the same amino acid. On fourth, fifth, they have the same amino acid. On sixth, seventh, they have the same amino acid. On 910, they have the same amino acids. So you can see at every position that I have marked in this table, they have the same amino acid in both. But every unmarked point actually represents a position in which the amino acid got changed, because the codon must have changed, and this codon change allowed a new amino acid to be placed. So this basically brings us microarray and bioinformatics. This is how they must have compared these using bioinformatic information. The two amino acid sequences shown in figure 3.1 can be compared. The number of amino acids that occur in the same position on both sequences can be counted and expressed as a percentage of the total amino acids present in one sequence. This is called percentage sequence similarity. So what is percentage sequence similarity? We'll just count the total number of amino acids. The total number of amino acids given to us is 32, and we'll count how many amino acids are common. So if we count, we see that total amino acids are 32. Amino acids which are common are 16. So we just take the percentage sequence similarity. It is already mentioned how percentage sequence similarity can be calculated. This information is relevant to figure out what formula you have to use. So you will just use amino acid common divided by total amino acid multiplied by 100. So that will give you 50%. So that 50% tells us that is the amino acid similarity. Compared to human calcitonin, salmon calcitonin is more biologically active. It means it remains stable in the human body for longer time, binds to calcitonin receptors more readily. Bioinformatics was used to identify more biologically active form of calcitonin to treat osteoporosis. Explain how bioinformatics help identify salmon calcitonin as a suitable form of calcitonin to treat human osteoporosis. So how this was done? So you need to recall the information of bioinformatics. So bioinformatics, we know it's basically having a database of stored information of nucleotide sequences of genes and amino acid sequences both, because these both are important when you are considering similarities between organisms. Of proteins pooled from all over the globe, this allows comparison to be made for the similarity in between two sources. The 3D structures of protein can be predicted too. The changes in the salmon calcitonin would have allowed a better folded protein than human calcitonin for better function in treatment of osteoporosis. The question continues. They are saying the salmon calcitonin is to treat osteoporosis made genetically engineered by the bacteria. The plasmid is cut and joined from new gene. The new gene is added. The recombinant plasmid is then introduced a bacterial cell. Name the enzyme that cuts the DNA plasmid. So that's restriction endonuclease or restriction enzyme. How the DNA is joined? It is joined using DNA ligase. Identify and explain two properties of plasmids allow them to use as vectors for gene coding. Plasmids are circular DNA and thus makes them more suitable than a linear DNA, hence more suitable for producing rDNA. rDNA is recombinant DNA. Plasmids have restriction sites on which the restriction endonuclease can act and produce a cut which are actually palindrome sequences. Lastly, they are asking you the concept of promoter, a very important concept whenever you are discussing genetic technology, because whenever you insert a gene, for example, this is the gene, this is the desired gene that we want to insert. Together with the gene, we must insert a promoter, because this promoter is the region which must get attached by RNA polymerase if the gene is inserted in a bacteria or in a prokaryote. And if it is inserted in a eukaryote, then the promoter must allow attachment of RNA polymerase together with transcription factors. Because in eukaryotes, you have two things things which must attach to promoter: RNA polymerase, the enzyme, and transcription factor is the protein. So you can just recall that RNA polymerase is your enzyme. This is the enzyme that attaches to the promoter region. And transcription factors, these are transcription factors. What are transcription factors? Transcription factors are proteins. And transcription factors are proteins, and these proteins, they also attach to the promoter, and both of them attach to promoter of a eukaryote. So promoter of a eukaryote. So whenever you need to express a gene in a eukaryote, the promoter must allow attachment of RNA polymerase together with the transcription factor, which are proteins. Just recalling the importance of promoter. The question here basically deals with the gene coding for salmon calcitonin is introduced in the bacteria using a specially designed plasmid called extension vector. An extension vector must contain a prokaryotic promoter such as lycopron. Why a prokaryotic promoter? Because a prokaryotic promoter is used because the gene is inserted in the bacteria. We are using bacteria as the in which we'll be adding the gene. Explain why difference in control of gene expression in prokaryote and eukaryotes mean that the expression vector's plasmid must contain prokaryotic promoter. So the promoter sequence of the prokaryote and the eukaryote are different. The promoter sequence are just the sequence of nucleotides. The enzyme RNA polymerase is also different. We know that they're both type of cells. So RNA polymerase of one would not recognize the promoter of the other, would not bind to its gene. The gene would not be expressed. Eukaryotic promoter require additional transcription factors to attach to promoter apart from the RNA polymerase. You can further say that the prokaryotic cell would lack these transcription factors because the transcription factors are already there are just there in the eukaryotic cells. That's it from my side. I hope that was helpful. Thank you so much.